Okay, so today we're going to kind of do the what's next lecture, which is a good lecture to have be last. So you have a phylogeny, you've measured, you have a phylogeny. We can show that this species and this species are different, right? And how different kind of depends on a lot of things. One thing that we kind of haven't touched on, mostly because it's pretty complicated mathematically speaking, is that these branch lengths here can often be the indicator of change. So the longer the branch length, the more different that sequence is than from the others. Now, that is dependent on a lot of things. One, that there's relative timing in your tree, and we'll talk more about that in a second. Remember that some of you are building trees right now, all the tips should line up if they're current living organisms, right? If it's something extinct, you would have something that looked more like this, okay? So now, later, right? But what I kind of wanted to do today is the more of the now what. Like, so what? You have a phylogeny, what are you doing with it? Why did you do this? Yeah, now we know the relationship. Maybe we've shown that systematics is more relevant than taxonomy, blah, blah, blah. But what else? So I was thinking about this whole week, and you're reading the Coevolution paper, and I picked these papers. Well, one, I did the papers all first. Like, what are some classic questions in evolution and classic papers in evolution? And I know for some of you would like to read more current ones, and I hope you see that I'm folding them in. And a lot of them are referencing current things that we're talking about, but maybe the research or the figures are better in this older paper, right? That's typically the case. And I just want you to get like kind of an iconic vision of what we're doing. And so one really classic study is an Ulrich and Raven study from 1964. And this was a study looking at how plant diversity is strongly correlated with insect diversity. They use arthropod, but honestly, they don't actually measure anything but butterflies. So we're not really talking about all arthropods here, but they're making this connection between plants and insects and how the diversity of plants led to this huge diversity of insects is the thought. Like, once organisms could become terrestrial, there were plants living on Earth, and then insects kind of came in and filled in all of the available niche space. Well, so that was 1964. We had an idea of DNA and all of those things, right? But we weren't really folding that into ecology type literature. This Ulrich and Raven paper is one of those top 10 papers from ecological studies. And what's interesting about it is they've now gone back and including in this study system exactly where these people sampled and done a phylogenetic analysis of the insects and plants that they talked about or cataloged in this paper. I didn't have you guys read this paper because it is long. It doesn't have a lot of figures. We just didn't get there. The humans and ticks is a lot more interesting and creepy crawly, and so that's what we did. But what we're looking at for this Ulrich and Raven paper, or the revision of it, is looking like this. So they built a phylogeny of the plant group, which is on your left, and then of the various insect groups on your right, and then looked at where you're acquiring these new traits, because things like mustard plants, and plants have all these toxic chemicals in them to keep insects from eating them, right? They're anti-predation. And then often an insect will be able to digest or metabolize, or whatever you would want to say, those toxic compounds so that then it can eat that plant. And it goes back and forth and back and forth. And this is called an evolutionary arms race, or coevolution. And what they found was a lot of times that wasn't necessarily the plants that were driving the insects. It was actually back and forth, back and forth. So I wanted you to look at something here. First, let's look at branch lengths. A lot of these, a lot of these branch lengths here are pretty shallow compared to these deeper branch lengths. So there were already, this is already showing that the plant groups here are younger than this insect groups here, because this is present day. So as you're going back here, this is ancient. And the same with on this side. I can't write the other way, sorry. Okay, so if we look at this, they're shallower here than they are here. So there's more genetic diversity in these insect groups. Also, if you look up here, there's a pretty similar, this new trait, that's what these stars represent, are new traits. Evolution of a novel anti-herbivory defense and counter-adaptation. So here's a new trait. It's actually newer than this new defense mechanism. One example of a defense mechanism they show here. So this calotropin is a chemical that should be really harmful to insects, therefore they maybe won't eat it. But what this insect does instead, or this caterpillar, is it is biting into this, building a little roundy moat here so that, building a little moat to trap all of the chemical defense, and then it's going to eat the rest of this here outside of the moat. So this vein can't shoot more of the anti-herbivory compound while it's being eaten. And so it's just back and forth, back and forth. This is coevolution. The humans and ticks, it's the same way. You see that as humans are diversifying across the planet, so are ticks, fleas, other arthropod groups. There's a good bedbug one also, which is just even, in my opinion, worse than ticks. Oh, lice is the one you guys are doing, humans and lice. And then you can also see that across mammal groups and ticks. There's a really good paper where it's tick diversity across mammals because of fur and hair differences cause different appendages and mouth parts. But this coevolution is really interesting, and a lot of times not necessarily a pattern you can detect until you start building these phylogenies and comparing across, okay? So that's what we're doing in this situation. You can also see this often in birds. They'll do bird insect or seed diversity. There's a classic paper that's doing Darwin finches the same way. So they built a finch phylogeny and a seed plant phylogeny. I don't know what kind of plants those are on the island. And they're looking across seed types and things like that. So I think what's interesting, and we've already talked about this some, is a lot of times if we take these classic ecological papers and we put them in an evolutionary ecology perspective, we get a little bit more knowledge of the universe. And that's the whole goal from an ecology perspective, is this knowledge of the universe. And so this isn't just true for medical fields, it's also true for evolutionary fields. Okay, so that was evolution. There is a link here. I'll post this PowerPoint. There's a link to a kind of cool blog about this follow-up paper if you're interested. Okay, so coevolution was like the theme of this week, and I was thinking coevolution, mutation rates, what else can you do with a phylogeny? And I was knowing that I skipped a serious one, and I just wasn't pulling it together until I read the human and lice paper again to write your quiz and gene flow. There are a million examples of when a knowledge of how alleles are actually moving in an environment completely changed what we understand about the study organism. For a very long time, most people considered songbirds to be monogamous, and they mated for life, and that is now the majority of those species that 15 years ago were declared mated for life actually don't. They're not monogamous. We know this because they started sequencing eggs and found that pretty much there's millions of ways in which birds sneak eggs and or sneak mating attempts into the system, and including like eggs that weren't even from that same species, which is interesting. But so that's a really good example of gene flow. Another good example, there's a cool hyena study where they looked at these hyenas have these packs, but they're female driven. So there's a dominant female. She has a hemipenis even, and she uses her pheromones to like control the rest of the pack. And so none of the other females in the pack menstruate while she's in charge. So she is the only one mating or successfully mating with these and reproducing with the males. And so the question is, like, how are all the males not so related then? Like, how do they keep this genetic diversity? And it turned out that none of the males that were with the pack, once they're hit a certain age, are actually related to them. They're swapping males from other packs. And so, I mean, we just didn't know any of that. Like, you knew that there was a lot of weird stuff going on with hyenas, but like how the behavior, like what the actual behavior is, because you can't ask a hyena like, how are you guys related and what's going on here? And it wasn't very observable. Once they did a phylogenetic analysis and they looked at how related all the individuals were within a pack than within a population, they really got a lot more information. And so these studies of gene flow are so cool because of these type of things. It also really, really guides our conservation efforts. And I'll talk a little bit more about that here in a second. I want to talk about the human and lice figure first. So there is this program. Well, let's start out with, so you build a phylogeny, again, as we always say. So you build this phylogeny. Oh, you'd think I would get better at this. Here we go. You build this phylogeny, and it's all like super related. Maybe even it looks like this because all the organisms are super related. And then you think, well, while this evolutionary knowledge is important, like maybe we're depicting it in the wrong way or asking the wrong question from it or something like that. What I really want to know is how populations from X place are related to populations from Y place, or individuals from Y place, okay? And so these would be Y. And then, like, sometimes you get these individuals that are popping into the wrong population or something like that, and you don't really know. So what they did was they used classic Hardy-Weinberg math, and they developed a program called Structure. And this literally is looking at the population structure of a community or a organism. And so what you do is you take a bunch of individuals that are all the same species, maybe from multiple populations or from a single population, or like two populations, depending on what your question is, and you throw them into this program. And so that's what they've done here with the lice. Sorry, I'm trying to get it to magnify this. There we go. And a lot of you are right. So this is figure 4. This is the one that the quiz is about so far. And those of you who haven't taken the quiz yet, you'll see it soon. So what they did is you throw all of these individuals in, and you have a certain number of markers, and the more markers you have, the more power you get in this type of analysis. So it just kind of depends on what you're using, but we can talk more about that later. And so you have all these individuals, and you're trying to get the computer to figure out where splits might be. So they're binning the individuals, and then they come up with a k value. And so in this case, it's k equals 4. And so they would have tested k equals 2, k equals 4, k equals 8, and those would have given pretty similar values probably, but one would be a lot higher. And then the odds, or the non-harmonic values, wouldn't necessarily, they would be crazy. And so it's a lot like model testing that we talked about previously, where the data, you fit it to a model. This is the same way. They're trying to bin these individuals into X number of populations, or K, it's actually K number of populations. And here they came up with 4 was the most logical. And so in this group, or this figure, their 4 populations come out here, and then, but those populations, or those groups, are made up of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 actual sampling populations, right? You want to sample fairly evenly if you can, because that can kind of alter your analysis. And so when you look at this, this is just a principal components analysis. People love these. I hate these, but it's pretty decent for this sake. You see that there's a group over here that's kind of bluish. Blue is New York, San Francisco, Florida. What's that red dot? And a random red dot. Green down here is Cambodia. Here's Nepal, but there's only 3 dots there, so that's not super informative. Orange is Honduras. Thailand's right there in the middle. We don't know what to do with Thailand. And then here's Canada up here. Oh, and these Nepalese lice are actually clothing lice, so it's a different species, so maybe we shouldn't have mixed those, but that's all very complicated. All right, so you threw all of these in to the structure analysis, and you came out with 4. That's super convenient, because if you look at this figure, you can kind of see that we can make 4 circles as well. Okay, and so here's Canada, here's New York, and the other kind of continental U.S. area. Here's Honduras, here's Nepal and Cambodia, and then here's some random other things, the UK, Norway. Again, here sampling is super low, so we don't really know, like, are these blue because they're coming from the U.S.? Are they blue and they were the original and the U.S. lice came from there? It's tough. But what a good example of what structure can do, and you'll see this again here in a minute, is look at this individual. So each bar is an individual. You guys did a good job with that. So this individual assigns 50-50 to Honduras and the U.S., meaning that its parents were likely from 2 different populations. So that's where you're seeing mixture. Thailand, you're seeing mixture. You would expect, here's this individual in New York, places that have a lot of in and out, right? A lot of traveling, like hotels or whatever, could see a lot more admixtures, or they call it, where you have individuals with assigned to many different populations, okay? The closeness here across the x-axis has nothing. You just rearrange these so that they all lie together and you can kind of tell what's going on. And then same with down below. Let's look down, oops. Let's look down below. Here we're looking at, what is this? Did I draw on this? More of this paper. But anyway, so structure will let you kind of get an idea of each individual's identity and where they assign to a population. Okay, so I'm going to give you another example. This is from some of my lab's work in Colombia. I came out of the Schul lab. We studied cone-headed katydids, and one of the reasons why we study these is because they're located in Missouri. They're huge, they're easy to collect, and we can find them in Missouri. And so in 2007, I started this analysis, and then this has been published recently by another member of our lab. And so what we did, essentially, can I blow this up twice? Nope, okay. Is we started here because this is Colombia in the middle. Sorry, you can't see the pointer. We start here and we sample, I'm gonna blow this up in a second, kind of all the way out. And then we sample over here, which has this giant barrier, which is the Mississippi River. Pretty classic barrier. And he kind of sampled in like a concentric circles all the way out, looking at the diversity of the populations, okay? So starting with Colombia and working our way outward. And let me tell you a little bit about what's going on in Colombia. So the blue, this is two different species that we're studying. They look identical. You could not tell them apart. I can somewhat tell them apart by call and by female shape, but in general, they can intermingle. So they can mate. I made hybrids. This can happen. The species boundaries are very, very loose, and they're mostly acoustic. So I made hybrids by having one female, having the right male call and the wrong male present. And so blue is one species and red is another. All right? Blue species loves prairies. You find these in prairies, pretty pristine, good quality prairies. Red species you find anywhere, especially in ditches. And so in Colombia, we would sample at a certain place for red and a certain place for blue, because the blue were in the prairie and the red were in kind of wetter areas, ditchy areas, right? But then we noticed that, well, we really could find many individuals in like of each species in each place. So that got kind of complicated. And then we noticed when we did genetic analysis of them, not just acoustic, like how were the males calling, that they didn't always call with the right, like they didn't always assign strictly to one species. So we were finding hybrids. One thing that this figure shows that isn't maybe necessarily clear is here on the right, this figure in B is not actually a phylogenetic tree. What it is it's called a splits tree. It throws out the window this rule that everything is bifurcating, as in you always split into two. So it just lets splits happen organically. Let me draw something. So you have bifurcating, right, is like this. I mean, and it doesn't have to be just because it's, but you always have A or B, right? Splits tree lets you have these, like, the idea that there could be multiple events occurring from one node. And this is particularly relevant because we're looking at individuals here. Each line is an individual, and those individuals could be siblings, right? So they could all come from the same mother. So that's why you have these clusters. And what we see is if you throw all of the robustus, which is the red, and all the bivocatus, which is the blue, into analysis, they do split. So you get most of the blue assigning to one side and most of the red assigning to this side. And there's a 95% agreement here that among all the trees that, or all the times we ran this, that this is a true split. There's two clades, okay? So this would mostly look like, if we did a phylogeny, something like this with 1 million of these, one for each of the red guys, and this. Okay? And so this is more informative. So that's what we did. And the letters that you're seeing represent the different areas. And so as we were sampling out, we kept doing structure analysis to see like, oh, well, what is this? Can you differentiate between one site and another site? And within this local area in Columbia, the first circle, no. And then we would move out to the second circle and then to the third circle. And then like, what if you sample across the Mississippi River? Are those genetically distinct from the ones that we've collected in Columbia or in Springfield or wherever? And so then we went to a structure analysis. And here it is. And oops, I see why this keeps happening. This first one goes with that little figure we were seeing earlier. And no, you just have a red group and a blue group, but you don't really get distinct groups between there. Somewhere recently, after a pretty big drought, which knocked out a lot of, or made us find less and less Bibocatus individuals, true Bibocatus individuals that were all blue, like here, you start seeing that there's kind of starting to be a split amongst groups. So you're getting more like a 3 to 4 population. Here it's popping at 3. And then I think I lost my ability to scroll. Amongst Robustus and Bibocatus, if we separate the blue group from the red group and then run it again, you get somewhat of a split, but still they don't assign these. Across the bottom are the localities. They're not assigning specifically to localities. There's some localities that have a higher signature of true Robustus or true Bibocatus, but there's a lot of hybridization. And so this kind of reframed, I want to go back to this figure here, really reframed what we said about conservation in Missouri grasslands. So part of this study was funded by the Missouri Department of Conservation to look at prairie remnants. So a lot of the places on this map are prairie remnants. So they're pieces of natural prairie that are thousands to hundreds of years old. Okay, and how good does a prairie remnant have to be? How many species does it have to have? All these different rules the conservation department needs in place in order to conserve these native prairies or to let fields that were once farmland go back to prairie. And so the general idea is once something's been plowed, that's pretty harmful to its seed bank. But burning is a lot better for prairies than plowing. And what we found for these Bibocatus, and actually there's another prairie species called Integer, and this is true for that species too. When it comes to Neoconchocephalus, which are pretty good indicators of prairie quality, you can have very, very small pieces of native prairie surrounded by farmland, and they will be found by the species. And they do serve as a good stopover point, but because there's a ton of gene flow, it's not like that little pocket is wasted or is never going to be found, and it does serve as a good refuge. So size isn't necessarily as important as just the more prairies that we can have, the better, was kind of our ruling when it comes to katydids. The thing about these katydids is they can fly really far. So that makes a huge difference in the genetic analysis and in the conservation plan. Okay, one last topic, mutation rates. So you have a phylogeny, great. What does that tell you about how fast the populations are changing? All it really tells you at some point is the populations are changing. But if you can add time in, you can look at how fast populations are changing, right? And in what ways. We were going to, for this class, read an Ebola paper that was great. from the Ebola crisis in 2015, but we didn't have time. The thing about that Ebola crisis was so profound was that the virus was changing so rapidly that it was pretty much shocking to everybody. Like, it changed, just making these huge leaps, which made it even more harmful than your typical Ebola outbreak. And how you know that is because you're adding molecular clocks. In the Ebola thing, they were doing it live. So they knew this was what the last outbreak looked like. This is what other strains of Ebola look like in other areas. And this is what, March virus looked like. And then the April virus. And then the most common May virus. And then all the way out to November. And they were looking at how fast it was changing. Okay, so your paper that you read this week also had a time tree in it, and maybe you didn't even notice. This is a tree that they built from mitochondrial DNA. So the CO1 gene, which is a common mitochondrial gene, it's in charge of metabolism, obviously, because it's in mitochondria. And what they've done is dated the nodes. So here are head and body lice from various places, and then they dated the age of these splits. Okay, and basically then they can look at how much genetic diversity has changed between this group and this group to figure out this age. All right. And mitochondrial DNA is particularly good for this because it's easier to get. There's more sampling. You can collect more off of blast. And if you don't exactly know the sequence of the gene that you're trying to look for, if you use a CO1 gene, they're a little bit more conserved in certain places, and then you can get the variation on either side. And so we commonly use these in phylogenetics. So I'm going to give you another katydid example of a practical working group. So a long time ago, when I first started my thesis, I built a phylogeny, which I forgot to put in here, but of katydids, and it looks something like this, blah, 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 da, 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 outgroup. Okay, and it has this temperate group, which are katydids that live in North America. And it has this tropical group, which are mostly katydids that live in the Caribbean. Costa Rica, north to Florida. And what we wanted to know was, when did these katydids that are almost all in a single clade, so all of the North American katydids pretty much live or are the most recent common ancestor to each other, and then there is a tropical most recent common ancestor. And so when did this split occur? Here. So we worked on this for a really long time, and like 1,000 different ways, and this is one of the ways we did it. And this one is cool because we haven't really talked about biogeography yet, and that's a really important part of phylogenetics. So a long time ago, there was a continuous wetland that happened between the coast and the eastern coast and the Great Lakes. So this was all marsh, like 10,000 years ago. Okay, and then the plates shifted and glaciers were receding, and what happened with that is the Mohawk-Hudson Valley started to drain. So then you got the St. Lawrence Seaway. The draining of the St. Lawrence Seaway made this area dry up, and so now there is no longer a continuous wetland between here. There's some marshland here, which has mostly been turned into farmland, and there's marshland here, which is mostly farmland, and then in between we grow corn or whatever. And so this was called the Mohawk-Hudson Outlet. And they first noticed this pattern because you would find certain species of plants in marshes along in the Great Lakes area, and you would find them along the coast of New Jersey too, but nowhere in between. And you wouldn't find them northern, you wouldn't find them southern, you wouldn't find them in other marshlands further west or south. And so that was interesting, and then they figured this pattern out. And so we can use, one, our knowledge of the genetic information in the katydid system that we already had a phylogeny for, and our knowledge of biogeography in order to date the rest of the tree, that split I was telling you about between temperate and tropical, by dating a node within here, and then I dated some nodes outside of Neoconchocephalus as well. So what we did was, well, I didn't do this, but Gideon did this. He collected a bunch of this species that we find in the marshlands here, and we find it here, so it has that exact Mohawk-Hudson outlet pattern. It's called Lyristes. He actually had to do this with museum specimens of Lyristes because he couldn't find Lyristes in the Great Lakes area. They have gone extinct, or a local extinction, we think. So here are the Midwest ones. Here are the Atlantic ones. We said that this split here was 10,000 years ago. And then what it did is it looked at the genetic differences between here, and it kind of does this like wobble thing. And it's trying to trace it back. How far back in time do we have to go until these individuals connect? Right? That connection should be 10,000 years. And then we can get this rate of changes per 1,000 years. Changes per million base pairs per 1,000 years. This is called a molecular clock. So you have a mutation rate, and then you can put that back into this phylogeny, and you know more about, or you can get some idea of when this temperate and tropical split is. I want to point out something that if you ever talk about molecular clocks in front of other scientists, you need to know. For a long time, people said the molecular clocks were not accurate. There are plenty of studies that say this, but what we now know is we have better math to do this, where you're letting the rates change along each branch at any moment. Also, you have to know the history of your study group and your taxa. And so what we did to make this is, I know that this is a temperate group, so it goes in here. I know a lot of information about everything else that's happening outside of here. I know that there's more genetic diversity in the tropical areas than there was in the temperate areas. And so what I found in the end was this split happened 12 to 20,000 years ago, which is as soon as the last glaciers receded. So at the same time that people were walking across the Bering Strait into North America, katydids were going up through Florida into the and through the Caribbean islands up into North America, because it was becoming grassland. The forests were receding, the glaciers were receding, and a lot of that was now grassland. Okay? All this area here has become grassland. So I did this by using molecular clock techniques in many different ways. like this way with fossils and this way with coalescence, which is what this type of analysis is called. Okay, hopefully that gives you a better idea of what we can do with a phylogeny. I'm sorry that this is long again. If you have any questions, please let me know, and we'll talk to you soon.